Good morning. Good morning. Good morning. Good morning. It's sunny. It's Monday. It's a ridiculously warm day in October, and we're going to share some enthusiasms for invertebrates together. So this is good. Thank you for finding. I see pigmentation, xerophilic, and cryptopodia, and gill in the bottom there. Let's take these off and continue. So, what are we continuing with today? We've been talking previously on Zoology 3700. We were talking about problems with excretion, of getting rid of the toxic byproducts of metabolism. That had a lot to do with salts and a lot to do with ammonia, right? And so we talked about different kind of organs, different kinds of the principal players in the nitrogenous play: ammonia, urea, uric acid, and guanine. And then we talked about whether or not you could be a conformer or a regulator, and if that was really just a dichotomy or in fact a continuum. And it's more of a continuum because most things in biology are. Because what is the most frequent answer to a question in biology? It depends. Now, today we're going to extend some of those problems. Now, we were talking about in excretion, we jumped from marine to aquatic to terrestrial in terms of where we drew our taxonomic examples to talk about strategies from. Today we're going to be talking about largely terrestrial stresses associated with water loss. Now, some of them have to do with, it's really kind of a natural continuation to think about problems associated with salts that you want to keep, salts that you want to lose, depending on the ionic balance and the osmolality of the solution that you're living in. But this is largely stresses that have to do with living in a terrestrial environment. And so we'll focus on one of the most successful terrestrial invertebrates, which is, of course, the insects. We're not going to leave them there, though. We're not going to leave it at that. We're also going to talk about isopods, which are still kind of insects if you consider that insects are just crustaceans on the terrestrial environment. And we're going to talk a little bit about gastropods. But cards on the table, it is a very insect devoted lecture. So we'll talk about strategies that insects have associated with meeting one of these principal stresses of their life in the terrestrial environment. So what is that? How does it affect their cuticle? How do we see adaptations like spiracles on the end of so we'll tie it into respiration. We'll talk about Malpighian tubules, which we picked up last week when we were talking about excretion, but we're going to talk about how adaptations in the Malpighian tubules associated with xeric living species in very dry environments are really associated with using a countercurrent system to making sure that you can retain as much water as possible. We'll talk about gills to lungs for taxa like the isopods that are most diverse in aquatic or marine environments. We'll talk about pigmentation and behavior and how that can affect water loss. We'll talk about other kinds of strategies associated with water that tardigrades use, and nematodes and trehalose, that kind of thing. We'll deal with some particularly incredible xerophilic adaptations, and then we will talk about community-level consequences. This is a bunch. We got 50 minutes. Let's buckle up and get started. This all sits in terms of the learning outcomes that I would imagine for this class for you is that you can identify and distinguish adaptations to counter water loss for sure. Synthesizing and explaining how water loss is countered in terrestrial examples that we talk about and expand from those into others that we haven't. Synthesizing and explaining adaptations to a xerophilic adaptation, because we're going to touch on that very dry desert-like adaptations throughout. And then think about how the physiology here scales up until the community and how they can affect invertebrate ecology, invertebrate evolution, and diversity. So for any terrestrial animal, their body fluid, any body fluid of a terrestrial animal, whether it's the snail, the nematode, the isopod, or that chelicerate, or you're going to lose water across any permeable membrane at anything less than 99.4% relative humidity. And it's really hard to find 99.4 relative humidity. If you spent a summer in Guelph, you might think, it's not that hard, Smith, but unless you're in a very wet cave environment, we are terrestrial animals. We are losing water. And resisting this or compensating for it, two different kind of ways of identifying the problem, water loss becomes a dominant factor in the environmental physiology for most terrestrial animals. And in a way that makes the terrestrial environment harsher, more harsh than freshwater or marine environments. So in a terrestrial environment, and you'll expand on this in lab, you have a shortage of water, you have ultraviolet radiation that's not being attenuated by as fast, as rapidly as by the medium that you're in, especially if you're in an aquatic environment where it can be all pretty much attenuated in the first several centimeters. You have temperature fluctuations that go up and down. We'll talk about that a little bit on Wednesday. You have obstacles to movement and the dissemination of offspring. You can't just kind of release your eggs, your gametes into the air because they generally just and they fall, right? That's kind of what happens. And so this is not under the sea. This is up on the land, and this is a challenge. The flip of this, in terms of me saying it's really bad being here, is that, of course, animal diversity is highest in the terrestrial environment, especially at a species level, not at a phyla level, not at the level of phylum. Like if we were to count tips of the Dunedin phylogeny, that's highest in the marine environment. But most animal diversity, most animal species are terrestrial species. That probably has to do with the diversity of habitats, and some of that has to do with isolation and fragmentation, as we unpacked a little bit in the first example in a marine environment with your copepods. But to live in the terrestrial environment means that you can solve problems associated with water loss. Consider the fly, right? If we take a longitudinal section of this fly and we draw on it in dark arrows where things are being lost, where water is being lost, and blue arrows where water is being can be absorbed. This is clearly a simplified fly, but because there are some taxa where we can absorb it, or they can absorb it through their foot pads and their palps. But water vapor can basically, that's WVA, water vapor aerosol, can be absorbed into the body through the mouth or through the anus, but you can lose it practically anywhere. You can lose it along your alimentary canal. You can lose it as an insect along your spiracles, so the trachea. Those are direct openings from the inside of the body to the outside of the body, right? So that's generally, even think about plants, like this is kind of a bad thing generally to have an opening of your direct body into the exterior environment. Cuticular water loss. So water loss, even where you have this exterior of the body solidified with nice kind of like beetle-like hard elytra, you're still going to be losing it across the cuticle. So you can ingest it, you can get water vapor at either end of your alimentary canal, but it is a big challenge. So ingested water is commonly transferred into the hemolymph or the blood across the midgut, so in this region here. So it happens very early on. It's valuable. Excretory losses, so losses associated with excretion, getting rid of toxins, are reduced in the secretion of less fluid by the Malpighian tubules. And you remember all those spaghetti-like blind diverticula, which in here are just shown as this horseshoe. So it's got, and there can be of the spaghetti that we talked about with the Malpighian tubules, dependent upon the species and likely how water stressed their environment is. Or water available their water is, how they have many Malpighian tubules, or they can have as few as 2. And then the enhanced recovery of solutes in water by the hindgut. Respiratory losses, so associated with respiration across the trachea and the opening from the trachea into the external environment, is called a spiracle, as it is in leaves and plants, mean that some taxa can control that. They can open and close, and we'll talk about that. Losses across the cuticle can be reduced by altering the composition of the lipids that cover the epicuticle. And we'll unpack some of this, the epicuticle, and we'll look at another cross-section and some SEMs of insect cuticle to just reflect upon how gorgeous insect cuticle is and how a lot more of your friends would be into insects if they could see the small and incredible features that you can see with scanning electron microscopy. And finally, in some species, reduced water activity on the mouth parts can permit water vapor absorption. Those are the blue arrows. Now, the other thing, if you think about insects, and you think about insects, consider the insect. most of life, so most taxa, you've heard me say the sentence before across a couple of years together, most of life is small. It's less than a centimeter, and it doesn't see the world, it smells and tastes it. So most of life is little. When you're little, and you remember this back to 2700 as well, there are some real stresses associated. So biophysical stresses associated with you that have to do with the differential, the scaling between volume and surface area. And we talked about it in terms of, you've talked about it in other classes in terms of heat, but we talk about it in terms of absorption and the appearance of microvilli to increase the surface area or increase the lamellae of gills to increase the rate at which you can bring in oxygen. One of the consequences of this ratio, the surface area to volume ratio, is that small things tend to lose water faster than big things. And this is a problem because most of life is small, most of life is insects, most of life is tiny and losing water. Now, I mentioned another drawing where we can talk about with a different fly. This is probably a Drosophilid, but in here, instead of emphasis, this is a glass half full fly, so it's emphasizing gains instead of losses. How do they deal with water loss? Well, they can visually detect it with their big eyes, compound eyes. They can detect humidity with their antenna. They can contact detect with their palps, the big parts, mouth parts. Some of them can do it via their legs or their ovipositor. And they can maintain and all of this is kind of, again, we talked about regulators and conformers. And oftentimes when people have introduced this false dichotomy to you in the past in physiology, perhaps it was in like ectotherms, invertebrates, insects, just conform. Not at all. Certainly not with water. So the detection, they have mechanisms for detecting and maintaining internal water homeostasis. The insect cuticle is where we're going to start. And this is a view into a tropical dry forest rove beetle. So rove beetles, there's at least 60,000 species of rove beetle. This is one family of beetle. They're the long ones that have tiny elytra. And that means there's more species of rove beetle than there are species of vertebrate. So we should consider them and think about adaptations that they have. In a tropical dry forest where seasonally it doesn't rain for months and months and months, there are likely to be a number of different adaptations from behavioral to physiological associated with conserving the water that you do have and fighting to prevent you from losing it. When we look again at that nice cross section, we've got the sensory hairs that were so evident in the last SEM. We've got this epicuticle, the exo, and the endocuticle. All of this is kind of being produced by these epidermal cells. And we're going to focus today thinking about the waxy lipid layer, this one, the epicuticle on the outside. It is, and we'll take this little tiny square here, and we're going to blow it up, and we're going to look at the fact that it's deposited through these minute canals, these pore canals in the exocuticle, and it's not completely waterproof, but it's very waterproof. Why is it not completely waterproof? That has to do with the need to get rid of balancing some of the costs and benefits associated. So you don't want to lose water, but you can't retain all of perhaps the ammonia and the excretory kind of byproducts that you've created. You need some of that to diffuse. And so you need a somewhat permeable membrane. And if we zoom in at a different kind of, like we've gone right in to looking at this waxy cuticle here. And you see the wax canals and that are kind of coming up through the epicuticle. And we have a lipid monolayer, and then we have lipid wax and cement, and then wax ordered. So disordered, what the little PowerPoint drawing is showing you here is that some portions of that waxy layer are laid down in a disordered fashion, almost like plywood, and some of them are laid down like you would see a hardwood floor in a living room, so they're nice and parallel. And they're accomplishing different things there. To unpack that, we'll need more time, but the diversity of waxes and lipids on the outside is the point here, that we have insects have a lot of them. The composition depends on the species, and the composition often depends or can be predicted by the kinds of environments that they live in, whether they're in a tropical dry forest where it doesn't rain for months, or a tropical cloud forest where it is raining all of the time, perhaps literally, historically. So another kind of a simplified, in this case, a beetle, because we were doing too much with the dipterans, but you're losing it. You're losing water across the cuticle. You're gaining it from your food. You're losing it from respiring. You're losing it from excreting, and you're gaining it from eating and drinking. So this all sits in this kind of thinking about water loss. This is kind of a hub in the center of a wheel that spokes out to other parts of the wheel, which are other themes that we've talked about. We've talked about excretion. We've talked about respiration, metabolism. So gaining water from eating and drinking. We're going to unpack those after Thanksgiving. But so water is clearly critical. And what I'd like you to do is to find, if you can, your annotation tool, because I'm going to ask you to make some simple predictions about water loss for these small diversity of the world, these small insects. And we're going to consider the y-axis here and say that is water loss. And we're going to vary temperature and ask, how does temperature affect water loss simply across the cuticle? So not the other ones, but let's just say, how does temperature affect cuticular water loss? So from 0 to 60 degrees, hypothesize. Nice. I don't know how you guys do it, but often you come up with different colors right away, which is impressive to me. And so the common theme, so well done. A theme right off the start here is that one that I would agree with, that it increases, that as temperature increases, you would expect to see more water loss. So I'm going to erase this, because I have another prediction that I'd like to make before we talk and collect them. So remember this. You predicted a linear increase. Whoops. Got to accomplish the goal first. There we go. Now what about humidity? How does humidity affect water loss? So we've got 100% humidity on the right-hand side, that deep cave environment, or the Guelph in August in the summertime, and 0, very xeric, very desert-like. So how does water loss, again on the y-axis, how is it affected by the relative humidity of the air to which the insect is losing its water? Hypothesize. Interesting. That's awesome. So some diversity in opinion here, but a consensus that the directionality of this opposes what we would see with temperature. Yes, I agree with that entirely. Some hypotheses that it might be more of a negative, what would I call that? A negative exponential, that there's an inflection point, rapid and then slow. Let us take a look at, so thank you. And this is, I'm going to take and unpack. So the prediction was that we have with humidity, or temperature, an increase, and with some variation with humidity, a decrease. And I'll tell you what we tend to see actually. Let's take out humidity because what happens with humidity is generally what you predicted. And that is that there is a linear decrease. So the higher the humidity, the less water that is lost, and that occurs kind of on a linear relationship. The difference and the inflection point that I saw in some of your humidity hypotheses, we actually see with temperature, and that has to do with the cuticle and particularly with the wax. So the aside here, I'll initiate it or remind you of it, that the cuticular permeability depends a lot on what species it is. And within a species, it can depend a lot on what life history stage you're talking about. However, in many species, we see little changes with an increase in temperature until a sudden catastrophic point. It's called a certain point there in the bullet. I'm going to call it a catastrophic point, where there is a distinct, that's a euphemistic term. This is a bad time, a bad temperature for this insect because past this point, right in here, water just begins pouring out. And that's likely due to the composition of those waxes, those either plywood arranged or linearly arranged waxes on top of the lipid monolayer that create the epicuticle, that create that barrier, that water barrier on the outside. And this change, this isn't something that and the reason I'm using catastrophe here is that this is not something that the insect recovers from. This is a permanent change. This is death. This is a big deal. So there's a resilience across a wide array of temperatures from 0 to 40 degrees. The exact point changes because the composition of the waxes changes from species to species. However, when this inflection point is reached, that is a big problem, and it might be the result is a rapid loss of water. And I'm going to hang this here and say, or I'm going to bring this point up again when we talk about temperature sensitivities together on Wednesday. So the cuticle, there are adaptations. So the waxes and the composition of the waxes are a big adaptation to still let ammonia diffuse out, to keep the water in. When you raise the temperature, you melt and disorder the waxes enough that the water leaks out, and that point is catastrophic. Now remember, one of the other principal ways that water is lost in our terrestrial insect is from respiration. So there's cuticular water loss, and then there's respiratory water loss. And this snazzy looking thing here, this is the opening to an insect trachea. And you can see that it is closed, right? So this is a spiracle that can actually be, water loss in this particular species can be limited by closing the spiracle. This is great if you're concerned solely about losing water, right? Because if you plug that hole, then the water stays inside the little round spherical insect. However, if this little round insect is wanting to do work, you need the spiracles open so that respiration can happen. So this is a problem if all the spiracles are open and closed at the same time. Insects evolution has come up with solutions to this. So let's take a look. So we've got the beautiful SEM here. This is not my SEM, by the way. This is not a Costa Rican animal, but we have where respiration is happening, we've got the tracheoles kind of innervating the the ner part of that is tripping me up, but they're innervating the cells right into the cells, into the trachea, and then we've got across this kind of oval spiracle the cuticle and an opening to the outside air. Now in some taxa we have what almost looks like those little feeding zooids from last year from bryozoans, the little like choppy ones to prevent that snap at the fish to prevent them from grazing. But this is, of course, this is a spiracle opening here. This isn't a zooid of a bryozoan. And what it has are closing muscles that can open and close the spiracle dependent upon oxygen needs and debt because of metabolism or water loss associated with the environment that you're in. So opening and closing is accomplished largely because of the innervation, literally the innervation of these of these closing muscles. Now opening them directionally, meaning deciding where they are, is another amazing adaptation that some insects have where they can open up. So we've got little spiracles. So we've got the light blue here is all of the trachea and tracheoles that go into the body from this insect. And you can see the openings on the side of the body. Now take a look at the arrows. Some of them on the abdomen are coming out, and some of the arrows on the thorax are going in. So those trachea on the thorax, that's creating a unidirectional airflow. So air comes in through these trachea and out through the other ones. And that directional airflow from thorax to abdomen allows some water conservation because the exhaled air is cooler in the abdomen, and so there's some condensation. Closing your spiracles is really efficient if you lock them all down. So we've got I'm going to unpack this plot here, but essentially we've got a flipped. So this is an important one. Instead of 100% humidity being at the large number at the right, 100% humidity. This is 100% humidity over here, and this is 0. So this is xeric, and we see spiracles open and we see water loss. So what's plotted here is an extreme water loss at zero humidity when the spiracles are open. As soon as we see 50% of the spiracles close, 70%, 80%, 90% close, you see a real reduction in respiratory water loss. And if you block them completely, you end up with this line at the bottom. So it's an effective way that evolution has derived to keep the insect respiring and to prevent water loss. So closing them reduces water loss, but interferes with the elimination of carbon dioxide and the gain of oxygen. And you think about this for animals that are flying in a dry environment. They need the spiracles open. So if they're forced to fly in a very hot, dry environment, there's a consequence to that, and the consequence is going to be water loss. Now, so cuticular water loss, respiratory water loss, what about through the gut? And we talked about the Malpighian tubules last time, and we didn't see anything as beautiful as this particular SEM. Now, this is an SEM through a gut of an insect that lives in a xeric environment. And I know this one lives in a xeric environment because of the counterchange exchange system that we see here. So we have the rectal lumen in the center of this star, and then we have lining in the star shape that I'm tracing out, this rectal epithelium that is then binding right beside the gut. So the rectal lumen, the gut, right? And in this case, in this organism, the Malpighian tubules aren't waving around like spaghetti. They are aligned and kept in track, essentially, by these epithelial walls right beside the gut wall. So the Malpighian tubules are not loose spaghetti. They are laid lasagna style right against the gut. So the fluid flowing through them is actually counter to the rearward, literally rearward, fluid flow in the rectum. So the direction of flow in the rectum is towards the anus, and the direction of flow in the Malpighian tubule is opposite, and that creates a classic countercurrent system that you've seen in biological systems all over the world, all over your experience of becoming a biologist as a way of increasing the efficiency of a transfer. What's the transfer here? So here, if you take a look just at this perinephric membrane, that's roughly where the cross-section would have been through that gorgeous star-shaped. We maintain our purple and blue and pink color module here, but we take a look. This is the rectal lumen down at the bottom, and you can see the dark arrows proceeding towards the anus. That's the rearward flow. Now we see the rectal epithelia, which is keeping, kind of lines the alimentary canal, but also in this case keeping along with the perinephric membrane, keeping the Malpighian tubules aligned not as loose spaghetti, spaghettini, but more as lasagna. And we see, I'm going to call your attention to some specialized parts of these Malpighian tubules. So first, before I get there, first of all, we've got direction of flow. So we have a countercurrent system here. Now, we also have this along the Malpighian tubules, these little lids, these specialized little covers or lids. These are specialized cells, the lephrophragmata, that tend to concentrate all of the potassium chloride. So one of the salts that potentially we'd be worried about excreting depending on the environment that you're in, but it's going to concentrate it here. So if we look at the numbers are showing you across the diagram on the bottom left-hand side. We've got 800, we've got 750, more than 2,500, but the highest concentration, the saltiest part of this whole process is right by these lephrophragmata. So that is a normal part of the excretory role that the Malpighian tubules are playing. The concentration, though, here in this cryptonephridial system means that those primary constituents of the urine, which are naturally there, they're creating water flow. And that's what we're seeing diagrammed here, is that so there's the countercurrent flow, but we also see a great differential in solute concentration. So this salty area here is pulling any water that remains in the gut contents out of it and saving it. And so that water in the gut is thus returned to the blood over that small osmotic gradient, which is kind of just to me beautiful and incredible xeric adaptation that we see in some of the tenebrionids or some of the insects that live in desert environments. All right, so we've talked about the cuticle, we've talked about respiration, we've talked about eating with the insects. I'm checking the chat and I don't see any questions yet there. We're going to keep going, and we're going to add some taxonomic diversity to our bouquet, our insect bouquet so far. And we're going to talk about some of the invertebrates that you stumble across on campus all the time, especially if you tend to go into the arboretum or the dairy bush and you turn over logs. You see snails, you see a lot of these isopod crustaceans. And there are some incredible adaptations that these taxa have, because for both of these lineages, the gastropods and the isopods, where's the vast majority of their diversity? It's not in the terrestrial environment, it's in the marine or the aquatic. So how are they making their way through life here? So I have a fondness for isopods. I love isopods. When I work in Costa Rica on the slopes of the volcanoes in northwestern Costa Rica, I cover an enormous gradient of temperature and precipitation, or humidity, a water gradient and a temperature gradient from sea level to cloud forest. And especially high up in the mountains, where we, coincident with finding a lot of water, we see a lot of isopods. And all of the leaf litter organisms eat a lot of isopods. This is an isopod here. This is another crustacean called an amphipod. But they're food. There are many lineages, many species of isopod around the world, but the vast majority of them live in the marine environment. There's only a couple of lineages, a monophyletic group of them, that has made the jump into the terrestrial environment. And they have 4 different kind of adaptations that tend to let them have let them take advantage of that terrestrial environment in the same way that some of their crustacean/insect other lineages have really done it with great success. So it has to do with the cuticle. It has to do with habitat selection. So we're going to bring in behavior now. It has to do with respiration, not this time in terms of spiracles a little bit, but not as much, but the difference between whether they have lungs or gills. And then the marsupium, which if you don't remember from 2700, I'm going to reintroduce to you because I love it. So unlike other crustaceans, terrestrial isopods do have a thin, waxy cuticle that makes them more or less impermeable to water. More impermeable to water loss than, say, all other isopods, less impermeable to water, or they lose more water than do most insects because they don't have as much of this wax. So they still live in danger of evapotranspiration, and this leads to your prediction or your observation of where they live, which tends to be, if you see them inside your house, you don't see them up in the attic or up in the main floor. You tend to find them in the basement or around the back of the toilet, somewhere that's moist, because they're losing water. They want to be in an environment where water is available to them. And so terrestrial survival depends on them finding that spot with an appropriately high humidity. Now, they do have some other incredible adaptations associated. So they have wax on the outside of their cuticle. This is independently derived from the from the insect lineages. So this is a solution, a waxy solution to living and dealing terrestrially with water loss. The other way that they have is they have what's called a biphasic molt. So when they molt, when they grow, when an isopod grows, you might have come across this before. The biphasic molt means basically they molt one half of the body, the anterior or the posterior, so not longitudinally, but anterior or the posterior. Part of that is to do with another limiting element in their life, which is calcium, which isn't as easy to find in some terrestrial environments than it is in others. And so they can, as they molt one half, they can actually eat that molt, the older molt, their older skin, to retain some of those elements that they've lost. But the other reason has to do with what we're talking about today, which is that new molt would lose water a lot faster than their older, harder molt. And so by going through a biphasic molt, it means they lose less water as they're growing. And there's an example here. So we've got the older, this is the shed exuvia back here. So they're going to eat this in a second to retain some of that calcium and other elements, but they're not going to lose as much water because of the biphasic nature here as if they molted their entire body at the same time. The vast diversity of isopods in the world respire via gills that are associated, like any sensible crustacean, with their legs, and you can see them down here. So we've got gills sitting in, but terrestrial lineages of isopods tend to have what's called a pseudotrachea, so a spiracle that goes into these pseudotrachea that open directly into the hemocoel. And we can see it there, that little almost like a fungal mat growing in here and here. So pseudotrachea are invaginations of the body wall that are better at conserving water than our gills, and they're more easily kind of erected and maintained than our gills in a terrestrial environment. And so isopod lineages that have pseudotrachea can tolerate and live in much drier air than can those with gills. So gills wasn't just lost coincident with achieving or arriving in the terrestrial environment. So there's the gill, here's the pseudotrachea, but this pseudotrachea is, again, just like the wax, this is not because of shared evolutionary, the shared evolutionary ancestor of the terrestrial diversity of insects with trachea and the isopod lineage with the pseudotrachea did not have a tracheal system. This is a convergently evolved adaptation to living on dry land. And we see that because isopods didn't just make the jump and it wasn't like contingent upon having these pseudotrachea, we see different lineages that live in drier environments that have more gill-like or more pseudotrachea-like openings. And so the drier the environment that they live in, the more you don't see gills. The more humid the environment that they're restricted to, you may see taxa species that still have gills, and taxa that have pseudotrachea. But in a dry environment, you would only see the pseudotrachea. And the fourth of the incredible adaptations is the marsupium. And the marsupium, next time you catch an isopod in the dairy bush, flip it over gently and look in between the coxa on the front. So this is the head here, this is the head here, and right in between those arrows, this is what's called the marsupium, which is a brood pouch where the isopod maintains eggs and the first instar larva. So the larva, they're kind of first, once they crack open out from the egg, they still live in this humid environment that their mother maintains for them. And so this is an example of extended parental care within this incredible terrestrial crustacean lineage. So isopods, we see cuticular adaptations against cuticular loss, adaptations against respiratory loss, behavioral adaptations that maintain them in a moist environment, and then parental care even associated with a life history stage that would be particularly prone to water loss, where they maintain them inside of the marsupium. Some other iconic things that you see out in the dairy bush include this banded snail. This is Sepia nemoralis that is out in the dairy bush. And as we talked about a little bit in 2700, they are famously known for estivating. So we're kind of focusing on some behavioral and some pigment adaptations here with our gastropod example. So this is tree climbing. This is a study done a bunch of years ago at UBC. So estivation, that basically is a senescence that can be invoked by low humidity. And it happens, and you see it happen in the dairy bush all the time. And in fact, you can go and explore different gigapens where there are snails that get caught halfway up the tree, and you can see them kind of nestled into the furrows of the white pine bark. And the snail has gone deep inside the shell, and it's laid out this mucus veil which attaches themselves to the plant but also then seals them off from water loss. And 2 days, even just having 2 days without rain, was sufficient to reduce the activity and increase the estivation. Now, one of the things about color loss, so this is a percentage of snails active on the y-axis, and then the time, so very quick experiment here. and where they drop the humidity below 55%. Snails, just as an example of this, so 2 days, but even like over minutes, snails become inactive very quickly when you reduce their humidity. Now, the difference here was that the yellow ones were active longer than the brown ones. Let's think about pigmentation for a second. In terrestrial snails, thermal location, so how hot things are going to get, is affected by shell coloration. So lighter shells tend to be found in high solar intensity environments, so like up top, really exposed. And so they're exposed not just to high heat, but also a lot of water loss stress. Darker shells found in lower solar environments. And so you can see here that across a continuum of the same species of snail that differed in its pigmentation, cartooned here, and this is still with the banded snail with Sepia nemoralis, from a nearly clear to a nearly dark, that the different wedge sizes demonstrate the activity at different temperatures. So that's our view through the dairy bush. I don't see any questions in the chat. So we're going to talk very briefly about anhydrobiosis before we move on to xeric environments. Because we're going to talk about it quickly because we've talked about these things before. These are your tardigrades, so some of you, this is your favorite taxon, that have a number of different mechanisms that allow them to live these incredible lives that minimize or avoid damage in extreme environments, including extreme dehydration. But this is more than just tardigrades. This is rotifers, nematodes, some crustaceans, and of course the tardigrades. And one of the ways that they do it is with a trehalose sugar. We talked about last year, but this is the incredible awakening of Sleeping Beauty, these SB1 and SB2, these two tardigrades from Antarctica that were reawakened after a large period of time in a collection by adding them back in to an aqueous environment. Now, how can they live this cryptobiotic anhydrobiotic existence? A lot of it, not all of it, but a lot of it has to do with a sugar called trehalose. And high concentrations of trehalose have been responded in a lot of these cryptobiotic forms, like Artemia, like this nematode, like the tardigrade. So tardigrades actually have, amongst this kind of specialized group, have some of the lowest concentrations. But trehalose has been shown to stabilize proteins and to preserve the integrity of membranes during dehydration. And that's thought to happen via a couple of one of two methods. That it either replaces water, so the trehalose forms hydrogen bonds with macromolecules present in the hemolymph and cellular structures in place of water, and it preserves native structures and prevents kind of the sharp slicing and deformation or denaturing that would happen otherwise with water. And the other is that the vitrification hypothesis, and that I'll simplify it just by saying that there's these amorphous kind of sheets of sugar are formed which protect the proteins and the membranes in the same way, or the result is the same. Caramel's reminder is not needed. We're going to move into the dry environment, and we're going to think about xerophilic adaptations. So not just all things, like all insects losing water, but what if you live in some of the driest parts of the world?
And there's a tiny black beetle that climbs up to the top of a dune, stands on his head, lifts his wing scales up into that flow. And on these wing scales, there are bumps. The tips of the bumps are like magnets for water. The sides of the bumps are waxy. So fog comes in, begins to aggregate on the tips. The water runs down the trough and into the predator's mouth.
That's amazing. These are tenebrionid beetles in the Sahara. And this fog basking, which is kind of looping through in this BBC video here, that might have to be cut out or blurred if you're attending asynchronously, but you can go and take a look at the link later on. We see it across multiple species. So ridges possessing bumps that are themselves very waxy, so that whenever the air comes in that has a fraction of water within it, the air hits those bumps, condenses, runs down these rivulets, and then the positioning of the body means that the water runs into their mouths. And there's variation across species of tenebrionids. So this is a family of beetle, but you can see in this one, if we take a look at its apex, you can see bumps and ridges that are going to, this is the posterior end, so it's going to be standing up and the water is going to be flowing down into its mouth. Now some beetles so this is a morphological and a physiological adaptation to preventing water loss or to rewarding water gain. And some of them have behavioral manipulations that even accentuate this, because some of these tenebrionid beetles trench, and they build these trenches to essentially use the sand to have the same effect on the fog that's moving past in the early morning. So that trench is going to, the bumpiness of the trench is going to condense some of the water droplets. That's going to sit in a more aqueous kind of an environment, available environment to the beetle, and then they can drink up some of that water in the trench that they build. So behavioral, morphological, and physiological adaptations to these xerophilic lineages of tenebrionid beetle living in very, very dry environments. We're all together. So finally, we're going to talk a little bit about how water loss. So we've talked about it right down to the cellular, right up to the organismal, and we're going to expand that to the community level. How does water loss affect diversity? How does water loss affect community dynamics? Well, think about, so we've kind of focused a lot of this discussion on insects. So this is where we're going to end up thinking about insects. But plant arthropod interactions, so grazing insects, are enormously important across the world. When in a hydrated environment, specialist insects can be more successful in wet conditions. Generalists tend to be more successful under dry conditions. Generalists also tend to move more frequently, tend to move farther, and tend to not be as specialized on the species of plant that they live. And so the likelihood of outbreaks goes up in a dehydrated environment. Water balance affects host parasitoid interactions. We've seen increased rates of parasitism on developing larval Lepidoptera or other kind of early instar insects during dehydration, where they're not successful. There's either signals that are being sent out that the parasitoids are more easily find the larva, or they're not as able to fight off the infection of the parasite. We see aphid consumption that is increased in drought. So we can see alterations in predator-prey dynamics. We see interacting with vertebrates here, that mosquitoes, so blood-feeding insects, that host interactions change when in a dehydrated environment. So blood feeding increases when this chicken is dehydrated. We've seen arachnids retreat and less frequently to prevent water loss, and the consequence of that, or one of the community level consequences of that retreat, is that there are more flies. Fewer things are predated by those by the webs of the arachnids. And we've also seen prey shifts. So in dry conditions, spiders that would have fed on to a diversity of taxa switch, and they switch their prey preferences to ones that have likely a higher concentration of water. All of these can trickle down into community and food web dynamics, and all are built upon water loss, consequences of losing water. All right. The terrestrial environment, it's a harsh place, but there are insects. The most successful lineage that lives here has numerous different adaptations towards dealing with the losing water, whether it's across the cuticle, across the respiratory system, or across your feeding system. We see different adaptations in different lineages, particularly fine and beautiful ones in animals that are associated with xeric environments like deserts. And there are consequences to this, not just at the level of the individual, like the extreme death at that inflection point. That's an individual level consequence, but that they spread out into the community. And Where are we going? We're going to, on Wednesday, talk about temperature and adaptations and ways that you would study temperature sensitivity and resilience to high temperatures and low temperatures. In lab, you're going to keep working on your class experiment. And as Shayla has noted on CourseLink, you want to make sure that you come to your lab section and do come this week, because the end result, the product of this, is going to be really built up upon in this lab this week. What else to work on? Well, way at the end of the week, your case study opens, and then if you haven't seen it already, your invertebrate R, the second assignment, is open, and the videos and the code are available, so give that a shot, and I encourage you not to leave it till the last minute. So, it's a gorgeous day. It's going to be hot again, which is terrible in the long term and enjoyable perhaps for you today, so I hope that you can enjoy it. Take care of yourself, take care of others around you. I'm going to stop recording in a second and check the chat. And if you've got any questions, hang around.